In this video, we will discuss about translocation of proteins. So before going to the topic, uh, it is uh, it is good to distinguish proteins um, in a way that helps to understand this translocation of protein topic in a much easier way. So proteins are categorized into two types. Which is related to translocation. One is soluble protein and another is you can say insoluble protein. Or the soluble protein usually soluble proteins has some some cleavable sequence, that helps to cleave or pass through the membrane, and it has around short hydrophobic ends at near and terminal. Short hydrophobic amino acids, you can say, near and terminal. Around 7 to 12 amino acids. And this insoluble proteins does not have any cleavable sequence, and at the same time it also has this hydrophobic ends, but these hydrophobic ends are slightly bigger than that of soluble proteins. These hydrophobic ends are also near hydrophobic ends, you can say, near, it's not exactly end, hydrophobic sequence, you can say, near and terminal. Around 20 amino acids, and this number is not fixed. It can vary slightly up and down. So what is soluble proteins? Soluble proteins are soluble in lumens, ER lumens, or in extracellular matrix of the cell. So these soluble proteins are usually usually secreted outside the cell or remain in the ER lumen, as it also has a cleavable sequence near the end terminus. So near end terminus. So this helps in the protein to pass through the membrane, and whereas insoluble proteins has hydrophobic ends, and it also has transmembrane domain. It also has transmembrane domain, and this transmembrane domain final destination is inside the or is at the membrane. It can be either ER membrane or the plasma membrane. So, and at the same time, it also has the longer hydrophobic sequence, around 20 amino acids. So all this will direct this kind of proteins to have their final destination either at plasma membrane or at any other organelle that comes in the process of formation of, or at the membrane of any other organelle that comes in between the process of formation of this protein. that is from ER to Golgi to TLN, TGN, Trans-Golgi network, and then to plasma, and then to final destination of the cell. And destination of cells. This is Trans-Golgi network. Here, based on the signature of the protein, it directs the proteins to its final destination. So, this is the major categories in which protein are categorized. Protein are separated to understand the translocation of protein. So, translocation of proteins are mainly occurred because of the term that is called protein. I'm not able to recall that term. That term is called protein initiation or protein translation. That term is called protein translation, or that has hydrophilic section in between, in the within the complex, hydrophilic section within the complex. So this in mammals, that complex is called Sec61, and in eukaryotes or eukaryotes is mammal. In bacteria or prokaryotes, this is called Sec-Y. This complex is a bigger complex, so it has been divided into different subunits based on its function, and is called alpha, beta, and gamma. So this alpha can span, or it can span the membrane 10 times. Span the membrane 10 times, and it's both N and C terminus is in cytosol, whereas beta can span only once, and it also has its N termini in cytosol, terminus in cytosol. And this gamma is also similar to beta. It is a single spanning protein, single membrane spanning protein. That has its N termini in cytosol, N terminus in cytosol. And what people have identified by comparing Sec61 and Sec-Y is that alpha and beta is more important, as they are more important in yeast or in bacteria. You can say in yeast and RK. They are more important in alpha and gamma section are more important in yeast and RK, whereas beta is not that much important. So from this and from other data also, they have they have made to a conclusion that beta is not that much important for yeast or an RK. And how they have identified that Sec61 complex plays a role in protein translocation. So it's not translation. I'm not able to recall that name. You can find that name from the reference paper that is there in the description. Or we can say, so let's come to the point. How people have identified this Sec61 why Sec61 plays a role in protein translocation. So what they have done is that researchers has conducted an experiment in a way they have used a protein with photolytic signal at membrane or transmembrane section of protein. And what they have identified is that during the process this protein acquire its final destination. It makes a cross-linking with gamma Sec61 gamma subunit in between. So from this this is the first time that people have this is the first experiment that people have done to find out whether Sec61 plays a role in the Sec61 plays a role in the translocation of protein. So from this they have concluded that Sec61Y subunit plays a role for translocation of membrane proteins. And for the time being, you can consider this can also be called as protein translocation. Okay, exact word you can get it from the reference paper. That's all for this video. Thank you.